Today's lecture is going to cover somatosensation. So somatosensation is actually any sensation that originates in your body, but we're going to focus on a particular aspect of the somatosensory system, and that's touch. You might not know who this person is, and if you do, kudos to you, but this is Helen Keller, who was an incredible woman. She was blind, deaf as well as mute, but was incredible in her capacity to be able to learn. She is touching the face of Dwight Eisenhower, one of our U.S. presidents. So, as I mentioned earlier, somatosensation is any sensation that originates from the body. But we're going to be focusing on the skin senses, so the sense of touch. But just so you're aware, proprioception, which is body position, is also considered somatosensation, as are any of the sensations originating from the internal organs. So butterflies in your stomach, stomach aches, those are also considered somatosensation, but we're going to be focusing on the skin senses, which include touch. So there are three types of skin senses, or senses detected by your skin: mechanical stimuli, which is touch; thermal stimuli, which is temperature; and nociception, which is the fancy word for perceiving pain. So we're going to be touching on the ways in which the nervous system transduces all of these different senses and where in the brain this information gets processed. So just like the sense of hearing, where you had mechanoreceptors in your ear, right? They were hair cells that were moving back and forth in response to fluid movement. Mechanoreceptors exist within your skin. They are sensitive to physical distortions. So they monitor contact with the skin, and each mechanoreceptor contains unmyelinated axon branches. These branches contain ion channels on the ends of them that are mechanosensitive. What that means is when the skin gets depressed or moved or modified in any way, it causes these ion channels to open. So gating, the opening of these channels, depends on stretching or changing in tension. Now, you don't need to know any of these receptors, but here are some examples of the ways in which mechanoreceptors in the skin can be gated by touch. So we have an example here where the skin gets stretched, causing the mechanoreceptor to open. Here's another situation where the mechanoreceptor is attached, or the ion channel rather, is attached to other proteins that get pushed and pulled, ultimately causing the ion channel to open. Here's another situation where pushing movement downward of the skin, right, shown here, causes the ion channel to open. So these are mechanoreceptors. They're not being activated by any particular stimulus other than pushing, pulling vibration. So these are changes in response to the skin being modified in some way. So these are different kinds of ion channels that can exist within mechanoreceptors within the skin, and there are many different types of mechanoreceptors that we'll talk about in the next couple of slides. So this figure is kind of like a slice through a chunk of skin. So not to gross you out, but this is the surface of the skin, what you'd see on the outside, and these are deeper layers of the skin, ultimately ending with a layer of fatty tissue. So these skin receptors, or cutaneous receptors, are located either deep within the skin or closer to the surface of the skin, and they respond to different qualities of touch. So Meissner's corpuscles, for example, right? They're more superficial in their presence within the skin, and they respond to light, touch, and pressure. We have Pacinian corpuscles, right, which are found in more deeper layers of the skin, and they respond to deep pressure and pressure changes. So there are a couple others that we'll also talk about in the next slide that aren't illustrated here, but they also represent cutaneous receptors. animation failed. So 4 cutaneous receptors are found in both hairy and smooth skin. So you probably didn't notice this, but other than the palms of your hands and the soles of your feet and your mouth, the vast majority of your body is covered in hair. So these cutaneous receptors are found in both smooth as well as hairy skin. So we've talked about Pacinian corpuscles and Meissner's corpuscles, but we didn't talk about Merkel's discs and Ruffini endings. These guys respond to indentation of the skin, and they're what is known as slow adapting. I'll get to that in 1 second. So the Pacinian corpuscles, right, respond to skin displacement, deep pressure, as well as rapid vibration, and Meissner's corpuscles respond to ridges of smooth, they're found in the ridges of smooth skin, and also respond to lighter touch or pressure, which is, so smooth skin represents the soles of your feet and the palms of your hand, and so they respond to light touch. These guys are more superficial in their placement within the skin. Both the Pacinian corpuscles and Meissner's corpuscles are fast adapting. So receptors that experience constant stimuli will end up adapting. This happens to every single sensory system: hearing, vision, taste. So it all they adapt with constant stimuli. So for example, right now there are clothes on your body that you don't feel because those skin receptors on your body have adapted to the constant pressure or touch of your clothes. So the Pacinian corpuscles adapt quicker to a sensory stimulus compared to the Merkel's discs and Ruffini endings. They are more slow adapting. It takes them longer to kind of stop responding to stimuli. And you want a difference in reactivity to sensory stimuli because, fast adapting or stopping responding to stimuli quickly is not necessarily evolutionarily adaptive, right? Because you never know when there's going to be an immediate change in the sensory stimulus. So Ruffini endings and Merkel's discs are slow adapting. They respond to the indentation of the skin. So sort of like pushing of the skin is what you would detect with Merkel's discs and Ruffini endings. So right now if you're sitting on a chair, the skin against your bottom and the base of your legs are slowly adapting to that sensory stimulus because that skin is being depressed by virtue of being pushed down against the chair. Okay, these receptors are encapsulated. What that means is that they're covered with some sort of tissue in a kind of like a protective way, but then you have free nerve endings that are just hanging out in the skin as well. So in the previous slide, they look like axons, but they are the free nerve endings, and they are typically found around hair follicles. They detect hair movement, but they respond to temperature and pain. So these guys are kind of ready to detect any sort of possible damage-causing sensory stimulus on the skin. So you can burn yourself, you can cut your skin, and these free nerve endings are the ones that respond to these two qualities of touch stimuli. So information from these sensory receptors end up going through nerves all over your body at different levels and enter the spinal cord. And we'll talk about what happens when they enter the spinal cord in a second, but I first want to tell you about the sensitivity that differs in these receptors across your skin. So the 2-point discrimination task illustrates the density of receptors that can be found in your skin. And by density, I mean how much these receptors are packed together. So the farther away the space between point 1 and point 2 illustrates a less densely packed receptor containing area of the body. The closer together the 2 points illustrates a higher density of receptors. So the reason why they illustrate density of receptors in the skin is because this task you have a person do with their eyes shut, and they have to tell you the distance between the 2 points where they can still discriminate between 2 points rather than perceiving one. So in the calf, for example, or rather the back, the 2 points would exist at 42 millimeters apart, where the person would experience 2 separate points. However, if you bring that point 41 millimeters apart, that person would perceive it as 1 point. This is not true for the thumb and index finger. Actually, any portion of your hand, those 2 points will be really, really close together before the person perceives it as a single point, illustrating that the density of receptors in the hands are much greater than the density on your back. And in a way, this makes sense because we gather a lot of touch information from our hands, our mouths, and our feet. I mean, little babies are constantly shoving things in their mouth and touching things with their hands because they want to gather as much sensory information about the object that they're holding as they can. And these areas of the body contain the greatest density of receptors. So information from the skin travels through nerves and enters the spinal cord. So the central nervous system is shown here in blue, the brain and the spinal cord, and the nerves originating from all parts of the body, including your big toe, end up entering the spinal cord before it gets into your brain. These nerves, right, from the sensory receptors join together, so the axons of the nerves are the axons of the sensory receptors form the nerves that enter the spinal cord, and all sensory information enters through the dorsal root, and the cell bodies of these axons, right, of these sensory receptors reside in a giant bulge right outside the spinal cord known as the dorsal root ganglion. Right? So they're a collection of cell bodies of these sensory receptors that have their ends all in the skin. So the dorsal root is the place that brings in all sensory information, and it first enters the dorsal horn. So the spinal cord can be kind of cut in half, and the top half, or the dorsal half, processes all sensory information, and the bottom half, or the ventral half, handles motor function. So the ventral root actually is going out of the spinal cord to target the muscles of our body, but we're not going to worry about that. Right now we're going to be focusing on the dorsal root, which are primary afferent neurons. They're bringing information from the body into the central nervous system, which then ends up in the brain and allows us to perceive some element of touch. So the spinal cord can be broken down into 30 segments. Each segment contains a dorsal and ventral section, so dorsal and ventral roots. The divisions are cervical, the top 8, thoracic, the next 12, lumbar, which is the next 5, and then sacral, which is the last 5. So the segmental nature of these spinal nerves, right? So I just want you to note, in case you didn't realize this, the spinal cord actually ends much earlier in your vertebral column than you probably thought. But what fills the vertebral column are the axons, or the nerves originating from the sacral and lumbar portions of the spinal cord. They traverse through these the rest of the vertebral column, but the vast majority of your vertebral column is filled with the spinal cord, but your bottom half is actually empty and just filled with nerves. It doesn't actually contain spinal cord. Anyway, the segmental nature of the spinal nerves and the sensory innervation of the skin are related. So what we can do is we can break down the skin into divisions or areas that are innervated by different spinal nerves that end up entering the spinal cord. So I'm going to show you an image, and these are the dermatomes. So the dermatomes refer to the area of the skin that's innervated by the right and left dorsal roots of a single spinal segment. So for example, C5, right, I'm going to go back. C5, which is right here, enters the spinal cord in this area, but it happens to innervate this area of the body. Now you're probably wondering, why is this information important? Oh, by the way, C5 is also innervates this area on the back. So why is this information important? So this information is critical to physicians and clinicians, particularly in the emergency room, because if a person has damage to the spinal cord where they have lost touch, right? So you can target or touch different areas of the body to determine where in the spinal cord the damage may have occurred without needing the person to go through an MRI. So knowing where in the body the spinal nerves innervate different areas of the skin allow physicians to determine where the damage in the spinal cord could possibly have occurred. So once the sensory neurons enter the dorsal root, they synapse. Some of it synapses with the dorsal horn, but it also branches off and heads to the brain. The white matter through which these axons that are traversing to the brain is called the dorsal column. And it's called dorsal columns because there's one on each half of the spinal cord. So there are two pathways that carry touch information to the brain. There are two major somatosensory pathways. The first one I've kind of pointed out to you is made up of the dorsal column medial lemniscus pathway. It's collectively one pathway, dorsal column medial lemniscus, and it refers to the white matter tracts through which information is flowing about touch and proprioception. This is body position. The other pathway carries information about pain and temperature, and it's known as the spinothalamic pathway or the anterolateral pathway, again based on where in the spinal cord it's traversing. So let's talk about the dorsal column medial lemniscus pathway first. So again, this is responsible for touch and proprioception. Information enters the spinal cord, traverses through the dorsal column of the spinal cord, and synapses with the medulla. From here, the axons cross over in the medulla. Remember, the opposite side of the brain controls the body, right? So left side of the brain controls the right. So in the medulla is where this information crosses and traverses through the medial lemniscus, white matter. It then synapses with the thalamus. Here, axons or neurons project to the primary somatosensory cortex as well as the secondary somatosensory cortex, and from here it goes to association cortices, but ultimately allowing us to perceive touch. So this is an image directly from your textbook. Again, touch information enters the dorsal root. Some of it synapses with the dorsal horn, but the vast majority of it enters the dorsal column, synapses with nucleus in the medulla, where it crosses and enters the medial lemniscus, right? That's why it's called the dorsal column medial lemniscus pathway. These axons travel up to the thalamus, where it synapses with thalamic nucleus, the thalamic nucleus ventral posterior nucleus, or VPN, and then from here information enters the cortex and synapses with the primary somatosensory cortex, which is what allows us to perceive touch, or somebody touching our foot. The somatosensory cortex demonstrates somatotopy, much in the same way that the primary auditory cortex demonstrates tonotopy. The primary somatosensory cortex demonstrates somatotopy. What that means is the primary somatosensory cortex, or S1, is organized into a map of the body. Each body part occupies a specific location in the primary somatosensory cortex, and this was discovered during neurosurgery by Wilder Penfield in 1937. So neurosurgeons are able to keep the patient awake, and so Wilder Penfield in 1937 opened up the patient's skull and exposed the brain, and the patient was awake because there are no pain receptors on the actual surface of the brain. So after local anesthesia on the skull and scalp, the Penfield started activating different areas of the brain and asked the patients that were awake what they felt. So in this way, he was able to map the cortical representation of the body, and here illustrates where in the somatosensory cortex different areas of the body are represented. So just to give you, to orient you, this is rostral, caudal, this is the nose, this is the back of the head, this is dorsal, and this is ventral, and the primary somatosensory cortex is located in the parietal lobe, a very thin, narrow portion of it. And so this particular diagram here on the right is a coronal view of the primary somatosensory cortex. So it's showing you that the face and mouth and even teeth and tongue are taking up way more space than things like the arm, for example. Same with your hands. They're taking up a much larger space in your primary somatosensory cortex than things like your back. But there is a map of your body parts in the primary somatosensory cortex. The primary somatosensory cortex demonstrates somatotopy, but as I'm sure you noticed, and I just pointed out to you, the map is, first of all, not always continuous, can be broken up, but it's not scaled like the human body. So the mouth, hands, fingers, face take up a much larger space in this primary somatosensory cortex than the rest of the body. You're probably wondering why, but the 2-point discrimination test that we talked about earlier in this lecture should give you a little bit of a hint. The reason why it's not scaled to the size of the human body is because different areas of the body have different densities of receptors and therefore can gather much more touch information than other parts of the body. So things like the hands are going to gather more information from the body because they have a greater density of receptors, and so they are going to take up more space in the primary somatosensory cortex. So researchers have created a somatosensory homunculus, which means a little man or little woman, to illustrate what our bodies would look like if they were scaled to their representation in the primary somatosensory cortex. So if we actually had a body that represented the amount of space it took up in the somatosensory cortex, this is what we would look like. We would have grossly large arms, humongous mouth and tongue, and much smaller other body parts. So the somatosensory homunculus is distorted because the greatest portion of the primary somatosensory cortex is dedicated to receiving input from those parts of the body that are capable of the finest tactile discrimination. In other words, areas of the brain that have the greatest density of touch receptors. So we're going to shift to talking about pain and temperature now, right? We've We've touched on, no pun intended, touched on touch, but now we're going to talk about pain and temperature. So somatosensation, in addition to those Pacinian corpuscles and Merkel's discs and Ruffini endings that are responsible for just touch, tactile information about nociception, which is pain, is also a critical portion of the somatosensory system, and they come from the free branched unmyelinated nerve endings that reside very close to the surface of the skin. So when these receptors get activated, they indicate to the body that it's damaged or at risk of being damaged in some way. So nociception is the sensation that's involved in the detection of pain. Pain, right, what we call pain is actually feeling or perception. What we perceive is pain. The stimulus that's detected by the free nerve ending is called nociception. So nociception is the sensation of the perception of pain. I'll let you think about that for a second. But nociceptors, which are sensory receptors found in these free nerve endings, can be activated, but the feeling of pain can come and go. They're distinct processes, right? So you might get a cut, a paper cut, and that skin remains damaged hours later, but your feeling of pain only occurs for maybe that half hour, an hour soon after the damage occurs. So the idea between nociception and pain are very different. They are distinct processes and handled by different elements of the nervous system. So I want to make sure that you know that pain, what we're calling pain, is a perception, and nociception is actually the sensation of the tissue being damaged. So let's talk about what activates these nociceptors, these receptors that detect pain. So strong mechanical stimulation, so a slap or running into a door, temperature extremes, right, going from a very cold into a lukewarm bath feels excruciatingly hot and burning, oxygen deprivation, as well as exposure to chemicals like acids or bases. There are a variety of nociceptors that exist and that respond to these different qualities of stimuli. We're not going to talk about them, but our ability to tell the difference between pain caused by temperature extremes versus a slap in the face is the result of different types of nociceptors in our skin. So nociceptor activation leads to the mechanical opening of an ion channel, much in the same way all of the other somatosensory receptors work. And then this information gets to the brain, but we can also have chemicals that are released by damaged tissue that can activate nociceptors. So when we get a cut, that damaged tissue releases chemicals that ultimately activates nociceptors and gives us the opportunity to experience pain, mercifully. So you've probably experienced this, but I'm going to point this out to you so you are aware of this. But don't worry about these fibers that are carrying information. Don't worry about this at all. It's not important. But I want you to be mindful of the fact that there are two kinds of pain that exist in our skin that we experience, that we perceive. There's the first pain that happens soon after the skin gets damaged. But after some time, we might experience the second pain that we might call throbbing pain that lasts a lot longer. So if you have experienced this, it's the result of two different kinds of nerves that are carrying information about pain, but there are two different kinds of nerves, one that gets activated at first pain, the sharp, ouch kind of pain, and then the other kind of pain that's dull and much longer lasting, the throbbing kind of pain, is carried by a different type of nerve fiber that ends up bringing it to the brain. All right, how does this information get to the brain? So pain information enters the dorsal root just like the somatosensory, touch information, but it's different because some of this information courses through on one side, but these fibers cross almost immediately in the spinal cord. So even though touch information crossed up by the medulla, pain information actually crosses in the spinal cord immediately and enters and heads to the brain through the white matter on the lateral area of the spinal cord, which is part of the reason why this is sometimes called the anterolateral pathway. Your textbook calls it the anterolateral pathway, but I'm calling it the spinothalamic pathway. It doesn't really matter to me, but you just need to know that there's a different pathway carrying information about pain and temperature than body position and touch. Okay, so this is the pathway that forms the spinothalamic pathway. Pain information from our skin enters the dorsal root, synapses with neurons in the dorsal root, which crosses over immediately, coursing through white matter before it synapses with the ventral posterior nucleus, the same exact nucleus that handled touch and body position, and information again heads over to the primary somatosensory cortex, where we are for the first time able to perceive pain. So information, we might draw our hands away from the pain before we even realize we've experienced pain. That's because information gets to the brain so much slower than information controlling our muscles. We're not going to talk about it in this course, but there are axons that project from the dorsal root directly onto motor neurons in the ventral horn that control that side of the body, causing it to pull it away. So rather than waiting for the pain information to get to our brain, we're like, oh, we're in pain, we must pull our arm away. This information is much faster and hits the skin, so we pull away much sooner than we even perceive that our tissue has been damaged. All right, so here is kind of an image of both of these two pathways that are carrying information about touch and pain. The dorsal column medial lemniscus pathway handles information about touch and body position. The spinothalamic pathway carries information about pain and temperature. But this kind of illustrates where in the brain the crossing is occurring. In the dorsal column, it's happening in the medulla, whereas in the spinothalamic, it's happening much sooner in the spinal cord. So I want to tell you something that might be of some use to you regarding pain regulation. So I'm going to share with you something called the gate theory of pain regulation. I don't want you to be concerned so much by this particular diagram, but I want you to think about a time that you might have like stubbed your toe or gotten a paper cut. You might have immediately squeezed that portion of your body, causing it to relieve some pain. There's something to that. That's not an old wives' tale. Touch information can decrease pain perception. Pain perception is gated. So even though our skin might still be damaged, our perception of pain can be lessened by touch. And this is exactly how it happens. So information coming into the spinal cord in the dorsal horn synapses with projection neurons that end up carrying pain information to the through the spinothalamic tract to the brain, right? But there are touch axons that also enter the dorsal root, that eventually enter the dorsal column, but some of them synapse with this interneuron shown here. This interneuron, let me erase all of these marks that I've made already because you won't be able to see anything of what I'm talking about. This interneuron shuts off, right, inhibitory, shuts off or shuts down or decreases the activation of this projection neuron that's carrying pain information to the brain. And this interneuron gets activated by touch carrying axons. So what this means is that when we experience pain, if we rub or touch or push that area of the brain, that area of the skin, it's going to activate these interneurons, which are going to decrease the activation of these projection neurons, which ultimately is going to result in a reduction in our perception of pain. Our skin is still going to be damaged in some capacity. We still might end up with a bruise, but our perception of pain will be lessened by touch. So the next time, you jam your finger in the door and experience pain, squeeze your finger and you will realize, or you will experience, reduction in pain perception. And that's entirely the result of the gate theory of pain regulation, that pain perception is gated. All right, this concludes our lecture on somatosensation. So somatosensation is any sensation that originates from the body, but we focused largely on the senses, or the type of sense that originates from the skin. Mechanoreceptors, or touch receptors, gather information about touch from the skin. There are a variety of receptor types for the different qualities of touch: pressure, vibration, pain. Different areas of the body have different densities of the receptors, which ultimately, results in that somatosensory homunculus we talked about later in the lecture. Touch information enters the spinal cord through the dorsal root and into the dorsal horn. As a result, different areas of the body can be broken down into dermatomes, which correspond to different nerves that originate from it. Touch information gets to the brain by two pathways. Dorsal column medial lemniscus carries information about touch and proprioception. Spinothalamic or anterolateral pathway carries information about pain and extremes of temperature. So the body is represented in the primary somatosensory cortex, with areas capable of greater touch information taking up more space, right? So this is somatotopy, but it results in the somatosensory homunculus. And lastly, pain perception occurs through nociceptors, but the somatosensory input can gate our pain perception. So that's it for this lecture. Don't forget to submit your reflection.